Okay, hello everyone. I hope you enjoyed the morning so far. My name is Caterina and my talk will be about assessing pulmonary concentration of inhaled aerosol. Hopefully my voice would not tremble a lot, so let's see. No good? Yeah, lovely. So briefly, the contents that I will touch during the presentation, I will give you like a brief introduction, try to explain the aim of this project, which is something that I've been working on recently. So it's just like quite early stage results, quite fresh ideas and experiments that are being performed, and the workflow. Okay, so just like a quick overview about respiratory diseases. Asthma, unfortunately, is the most common chronic disease in children. Lower respiratory tract infections and pneumonia kill over 4 million people every year. COPD affects over 200 million people globally, and I guess there's no need to mention COVID, but recently the WHO says that we are out of the pandemic, which is great. The numbers around respiratory diseases are huge. So they account roughly 10 million, 14 billion in the EU and 11 billion pounds in the UK is the cost that we need to account for treating respiratory diseases. And the situation is not predicted to get any better due to air pollutants and I don't know, hopefully not other viruses that can circulate, but that is a real possibility. So I just wanted to show you what a chronic disease in our lungs actually means to us. So this is the representation of healthy lung and the lung of a person with COPD. So we get exposed every day to smokes or noxious particles, pathogens, and they trigger our immune system. So they trigger the response of macrophages, eosinol, neutrophil, and they all activate endogenous components like inter-EL, that stands for interleukin, TNF, tumor necrotic factor, and they all cause inflammation and remodeling of the lung tissue. And this causes lung damage. So, for example, emphysema, small airways disease, and chronic bronchitis. So, what I've been working on recently is trying to assess the salbutamol in tissue, and I will try to explain why salbutamol. So what is the purpose of this project and how are we going to face this problem is two scientific questions that I'm asking myself at the moment. So what I want to do with this bit of the project is trying to assess salbutamol retention within the tissue and established, so how we're going to face the problem, I will establish an LCMS method to do so. And what I did is quite recent, quite early stage. But I wanted to say, why salbutamol? So salbutamol is accounted as a short-acting beta agonist. You can see the chemical structure on the left, but I also wanted to give you a sort of like real-life image. This is not a product placement. I've not been paid by GSK, so no offense to our sponsor, but it's possible that you see this like a child using it, or I don't know, it's quite common, especially for treatments of asthma. Is quite useful because they have like less side effect compared to LABA and ICS. LABA stands for long-acting beta agonist, and ICS stands for inhaled corticosteroids, and they are normally the treatments for COPD. But unfortunately, they also get along with so many side effects. So since COPD is a chronic disease, clearly the treatment goes into like a long-term exposure. And ICS actually in the long term also can lead to osteoporosis, which is not very pleasant. And LABA also gives side effects like hypokalemia. So it's like a depletion of potassium in your systemic circulation, tachycardia, tremors in your hands. So this is not like an ideal treatment if we need to think in the long term. And another fascinating thing about salbutamol is that it's quite, we can say, tunable molecule. And it is reported in literature that it can form complexes with polymers for controlled release systems. So they can be encapsulated into chitosan nanoparticles, PLDA, and this actually mean like a new drug delivery systems for salbutamol. A little bit about the biopharmaceutics of salbutamol. So it is reported that the cellular transport involves organic cation transporter, specifically OCT3, that goes under passive diffusion for molecules that are positively charged. Or it can also pass through the tight junction barrier, which is what we see in our lungs. So our lungs are characterized by a fine air-liquid interface, and the presence of tight junction is essential. So what it does when it actually crosses the airway epithelial cells is that it links to the smooth muscles, and it actually allows the opening of our airways. And about the pharmacokinetics is that upon inhalation, the salbutamol produces bronchodilation within 15 minutes, which is ideal if you have like a sudden asthma attack, and it lasts for 3 to 4 hours. What is not ideal is between 58 and 70% of the dose actually is excreted in the urine over 24 hours, with 60% of the drug, which is in the inactive form. So we have a very good molecule, but when it's actually inhaled, we are not really leveraging the power of the drug that we have at the moment. Okay, so this is more or less the workflow. I decided to put a diagram because I'm pretty sure that you're not keen to see like the rat lung tissue cut, which is not very pleasant to do in real life. So I just put a diagram. So what I normally do is take pieces of rat lung tissue, homogenize it with PBS, and then I normally produce 2 sets of samples. One when I put the analyte, so the salbutamol, an internal standard, and the matrix tissue, and they are all centrifuged together. And then I produce another set of samples with the matrix, and then I add the analyte and internal standards afterwards. So I can actually calculate the recovery that I can get out of the tissue. And then like the samples are treated with acetonitrile, and then they go directly into the LCMS, which is my bioanalytical technique. So the LCMS is the tandem between liquid chromatography and mass spectroscopy. And what happens is that a stationary phase, there is a mobile phase that runs over the stationary phase. This divides the compound, and then they are directly injected into the mass spec. This is normally divided into like a mass analyzer. Oh, no. Oops. So we have a mass analyzer, which what I'm using is a triple quad. At first, it is ionized with a technique which is electrospray ionization. So it's a quite mild technique that will not affect the salbutamol. And then it goes into the triple quad to be analyzed. We characterize the different fragments, and then the detector gives the results in terms of mass to charge ratio plotted against the intensity of the signal. Yeah, so this is the results that I got from my LCMS analysis. I plotted the chromatogram of my salbutamol at the LLOQ, which is the lower limit of quantification. And in my case, what I achieved is 25 nanograms per mil. And the signal to noise ratio, the guideline says that it needs to be above 10, which is good. Both my salbutamol and salbutamol D3 is my internal standard at above 10. Why I am, even if they are like more or less eluted at the same time, I'm quite sure that I'm treating 2 different molecules because I'm monitoring different channels. So I'm monitoring 2 channels for salbutamol and 5 channels for salbutamol D3. So that actually allows me to say, okay, this is my analyte and this is my internal standard. I mean, you can see like a beautifully linear calibration curve. The results are plotted as the RRF, which is the relative response factor, and a ratio between the area of the analyte and the area of my internal standard. And this is the results that I got. So as I explained before, the recovery percentage, I got it from my 2 different sets of samples that I prepared. So I actually know how much of salbutamol stayed in the tissue and how much I can actually recover. In terms of accuracy and precision, they are given as percentages. And the guidelines that I'm using is the one from FDA for bioanalytical methods. Why the guidelines from FDA? Just because they are the most as strict as you can get. So if you fit into that guidelines, then you're more or less sorted. So precision values, they need to be below 20%, and accuracy, they can span from 80 to 180%. So plus or minus 20% from 100. So conclusion, what I did so far is try to adapt an LCMS method to quantify the presence of salbutamol in tissue. What I have to do next is try to validate this method. And the exciting bit is that we can also investigate possible complexes that the salbutamol can join in with other molecules and with other polymers. And this could actually provide a new therapy perspective for COPD patients. Unfortunately, I can't disclose which kind of polymers complexes I'm going to form because it's too early and because I'm not allowed. But yeah, this is it. And I would like to thank my supervisor, Prof. Daramonain, and the Dr. Daniel Baker, which is the postdoc that is providing endless support to me, or the team at the CDT Aerosol Science, because they're very kind and lovely for whatever, and EPSRC and UKRI, they offer me a placement at the government, so I will work and see how science can shape policy and work with policymakers and policy institutions to make informed decisions. So yeah, that's it.